That's probably going to be the most popular, so I gave you guys lots of room here. And both sides of this Xerox sheet. It's essentially my lecture, really. All these parts, you know, neck 3 petal, sepal, stamen, ovule, stigma style, right? There's the anther sac, filament, developing pollen grains. So it should reinforce the lecture. Number 2, remember we had that lecture, actinomorphic, regular dicot. That's a stone crop. 10 stamens, 5 petals, 5 sepals, good monocot, good dicot numbers, 5s. Everything's in 5s. Okay, see the 5, you even got 5 pistils. That's unusual. And it's the yellow guy right here. They're all in order. Okay, you see the 5 pistils? And I think I took off this part here so you can see the seeds inside, several seeds. Okay, and then we have be careful, this one fall apart, so don't try and pull things off of it. Number 3 is the zygomorphic, bilaterally symmetrical flower. Okay, I took off one of the wings here, so you can see the folded keel. There's one plane of symmetry, there's little peas inside the ovary. Okay, and then finally number 4 is the monocot flower. That's tulip. That's a sepal. How do I know? It's on the outside. That's a petal. Okay. So sepal, petal. 3 of each, good monocot numbers. 6 stamens, 6 times 2, 3 times 2 is 6. And the one, you can see 3 blue stamens there. There we go. Trilobed ovary is tricarpellate compound. Those are the seeds inside. Okay, now let's get to the slides. Now I know I have 5 and 6. 5 are, we have the apple flower here, you can take a look at the models. And 6 is the ryegrass with the glooms and limas and the stamens sticking out. But I don't have any slides of that. Number 7, like I said, number 7 is a lily flower, and we're running a little short, so just 1 flower bud for every 2 people. So for every 2 people, the lily flowers are in this preservative. They are huge, okay? Don't need no stinking microscope. Just pull it out with your bare hand, put it on a piece of paper, and open it up. You'll see 6 stamens, you'll see a pistil. I mean, the pistil is like about 3 inches, 3.5 inches big. So it's huge. All right, now our first slide actually is number 8. And this is a flower bud through lily. Here you can see the trilobed style. And the anther sacs. These are sepals and petals. They all look like leaves, but they are. Okay. But look in the anther sacs under 10X, and you can see the pollen grains with how many? 2 nuclei. A generative nucleus and a tube nucleus. The generative nucleus will make 2 sperm after pollination. Okay? Now, the next 3 are a unit. Number 9, superior ovary, hypogynous flower. See all the stuff is attached where? At the base of the flower. See the tomato seeds? Okay, ovules. So the superior ovary, hypogynous flower. Then for epigynous, number 10, we have apple. Here's some seeds, but you see how all the stuff is attached where? On top of the ovary. So it's an inferior ovary, right? And then the weird one, number 11, perigynous, there's a floral cup that actually goes around, you know, in and out of the blackboard here of this cherry. And here's the pit, or the one developing seed inside. So everything is attached here around the lip of the cup, you see. And here's the nice style and stigma. Floral cup is good enough. Okay, then we have one more photocopy for you to get, and that's over here. Okay? It's a 11 sequence thing of the development of the female gametophyte. That's number 12. There's the first 3 models. There's a nice ovule. Okay, that's 1. A megasporocyte. Under going to go meiosis. What do you get from meiosis? 4. 3 of them die. This guy, a megaspore, enlarges a little bit. There's 11 models, okay. Division 1, Division 2, okay. of mitosis, I should say. And then division 3. So how many cells do we have? 8. 8. Okay? Remember what they are? 3 antipodals, 2 polars, 2 synergids, and an egg. And finally, the last one puts it all together for you. There it is, the embryo sac, the female gametophite. There's a nice pollen tube that's yellow, coming down, through the micropyle to fertilize the egg. How many integuments? 1, 2. 1, 2. Micropyle, ovule. The base there, the whole big thing with the ovary. The whole slide is one pistil. Okay, back to slides again, huh? Number 13. Now, you're going to have 4 cross-section ovaries on your slide, times 6 ovules. So you got 24 ovules. Of the 24, how many are good? If you're lucky, maybe 3, maybe only 2. So, garbage, garbage, garbage. These all look like garbage. Is there one with 1 cell somewhere? I can't. They all look bad. Well, that one doesn't look too bad. Higher power. There we go. I was right. There. See? That's the megasporocyte or megaspore mother cell.